Hello, Richard Veale from Kyoto University. This is work with Chiang Chin and Tadashi Issa, also at Kyoto. And today I'm going to talk about whether marmoset monkeys model human and infant gaze. Preemptively, I'm going to be clear, there's no new infant data or even analysis of infant data in this paper. Rather, we look at other studies, including infant data, compared to adult macaque and marmoset monkey data to establish whether there's a similar pattern in behavior between the infants and one of the animal species. We have actually started collecting data from the infants to directly compare with the animals to basically answer the hypothesis or theory that this paper posits, but that is still not complete yet, and we didn't write about it in the paper. But I'll give you a teaser at the end. Our goal is basically to understand gaze. What drives a baby to look at one location versus another, or any animal for that matter? To really understand gaze, we need to quantify both behavior and also how the brain causes that behavior. With regards to human infants and, of course, human adults, there is lots of research recently happening in behavior since we have cheaper and more easily accessible eye tracking technology now. But there still is a dearth of research into the brain. And this is, of course, because it really is just not possible to do really controlled brain research with human infants, because such controlled brain research almost always is very invasive, involving surgeries, opening the brain, causing selective lesions, and things like that. So, as with human adults, we really needed a good animal model to probe the gaze attention system of human infants. For human adults, the macaque monkey has been a traditional and very successful model of the human visual ocular motor system. Is because the macaque monkey has similar eyes, similar photoreceptors, similar visual cortex, similar whole visual system, similar eye control, ocular system, etc., etc. They also, of course, use their hands for fine grasping and things like that, so it's good for other reasons as well. They make similar eye movements in terms of the quality and the quantity of them, and that's been reported, of course, in our previous paper and many papers before. Our question really is, what would be a good model for human infants' gaze behavior? We know that macaques maybe are a good model of adults, but what about infants? Would macaques or baby macaques be a good model? What would be a good model? In this paper, we look at different papers that review different subsets of human adult, macaque monkey, human infants of different ages, and marmoset monkeys that are freely viewing video stimuli, and try to bring together basically the analyses in these papers to compare them so we can compare apples to apples. The different papers use different methods, different stimuli, different analyses. We try to compare them as closely as possible. In our paper, we have lots of tables like this looking at, so for example, the different study, what type of animal and how many they compared, and what type of analyses they did, what type of videos they used, whether they were color, whether they had sound, etc., how big they were. You can see that among the different studies, lots of things changed. The size of the stimuli, the videos used, etc., etc. So it's understandable that maybe some of the results would be different. But it turns out they're actually more different than we expected, and so we suggest some better ways of analyzing data to bring them more in line. In the papers, basically, there's two types of analyses that we look at. One is gaze similarity analysis between subjects or species. The other one is gaze similarity to a model. In this case, a bottom-up attention model called the saliency map model. And so basically, I'll cover the results very quickly in two parts. First, I'll talk about the intersubject correlation one, comparing between subjects. So basically, you can correlate gaze as you'd imagine. You have different points in times of a movie where a subject is looking at a different location, and you can have a different subject looking at a different location, and then you can basically correlate those while they're looking at the same video at the same time. How similar are the places they're looking? So here you can see gaze data from this video, all the different subjects and the different animals that watch this video, where they're looking at each time point in the video. Maybe it's hard to see because it's so fast. I can slow it down 3 times so you can see more clearly that they're spending about, 300 or 400 milliseconds looking at one location and then making a usually saccadic eye movement, a fast eye movement to a different location. There's no macaques in that example, just no macaques happen to watch. Here's some example with macaques also involved. The reddish colors are macaques. Another example of some from the video database we used in ours, looking at humans, macaques, and marmoset monkeys. You can see they're making varied eye movements around the videos, watching the videos. Some more examples. You get the idea. Basically, you can plot these out as the X position of the gaze and the Y position of the gaze on the video, and basically correlate these traces between two marmosets, between two macaques, between every two pairs of humans, and take the average of that. And you get what's called the intersubject or within species correlation, the averages. And so you notice for marmosets it's very low. For macaques it's somewhat higher. This is still 0.13 or something. For humans it's higher, but it differs a lot between the different studies. The different shapes here, circle, square, triangle, are different studies. So all the studies looked at human adults, and you'll see that some of them reported very high as 0.6, some of them low as 0.2. There's some data from human infants here, only from the one study, so can't really compare to other studies there. In the paper, we have lots of data looking at the results where available between subjects, between species, things like this. I'm not going to cover it. A better way than the intersubject correlation is to use what's called inter-observer model, which is a model-based approach where basically instead of correlating gaze directly pairwise, you use the gaze of all the other subjects to predict the gaze of this subject at each point in the video. This is advantageous because we can actually use a better null model, or a model that better corrects for the prior probability of the subject looking at a given location to give a sort of more fair estimate of how good our model is. And then to quantify the performance of the model, we use the area under the receiver operating characteristic, AUROC, which basically traces out true positives versus false positives and gives you an idea of how predictive the model is. Here's a table showing the data. Basically, you can see here humans are inter-observer model 0.72, 0.61, 0.55. Inter-observer model 0.5 is chance for AUROC. And so these are not as high as they might seem. 0.5 is chance. And they go from 0 to 1.0. You'll notice that there is still some difference between the different studies, which is worrisome, but of course these are only two studies. Low ends, it may be due to chance. Next, I'm going to talk about the bottom-up saliency maps. So to remind you, saliency, basically you have some video input, some visual input, and you have separate parallel processes that filter for color differences, orientation, local orientation differences, motion differences, or luminance differences at different spatial scales, different sizes. And then those are all combined together into a final saliency map that predicts the position of looking based on sort of a feature agnostic in a feature agnostic way. It doesn't care if it was movement that draw attention or color that would draw attention. It's just a single saliency map. And then again, we can use AUROC to get the gaze. We use the green colors here. Cyan is where the subject is actually looking. The the magenta is where they could have looked, so they're drawn from their prior probability of looking there from other trials, other videos. And you do the false positives versus true positives, get the area under that, and that gives you an AURC. For all the species, all our subjects, they had an AURC of roughly 0.6 for the saliency map, implying that all the species are drawn by saliency at roughly the same rate. And so you can see some drawn just the same rate regardless of whether you use percentile, which was used also by the infant study, and whether you use AURC, which was used in our study and also in Berg's study. Notice that for some reason, maybe it's the percentile method, but the infant study, which only used one video, has a significantly higher saliency percentile than ours, maybe due to how they applied the saliency map, how much gray there was around the edges or something like that, might give it bias it up or down, which is why AURC is really a better method of quantifying that and comparing between studies. Here's the numbers if you care. Finally, we want to, this is really the meat of our paper, is the novel analysis. We want to understand whether saliency is actually affecting the variance of gaze and whether that might be a hint for why marmosets or infants are different from adults. And so basically what we want to do is reconcile this difference. Our saliency, we had roughly equal saliency predictivity of all the different species, whereas the inter-observer within species are different. Humans had a high inter-observer. They predict where one another look very highly, whereas marmosets very weakly predict where one another will look. And so this is important for saying, are macaques really a good model of adults? Are marmosets really a good model of adults? Maybe what's a good model for infants? And you can imagine sort of two hypotheses. One is that all the species look to the same salient things, and the variance, in other words, variance is what causes this, right? Inner subject variances. The variance comes from time points other than those that have saliency drawing the gaze. So in other words, time points in which they're looking to not salient things. They're looking to different not salient things. Hypothesis 2 is that basically they're looking to salient things, but they're looking to different salient things. And so basically we want to compare between these two hypotheses. And to do that, we quantify using what's called between subject gaze error. And in this case, basically you have two different subjects looking at different, maybe or the same locations at the same point in each video. You measure how far apart those are for each point in the video, and then you compare saliency as a function of that distance. And so you can see that here is a human example, macaque example, and a marmoset example. And you can already see that maybe there's a qualitative difference between the marmosets and the macaques. It's sort of like the marginals are way more spread out for the marmosets than for the macaques and the humans. And we can sort of get that more quantitatively clear in using a cumulative density plot. So basically here we're plotting the density of how many looks were at a certain distance between the two pairwise distance between the two subjects as a function of distance here, right? Oops. And actually I've separated this into high saliency, in other words, greater than the mean saliency, or low saliency, lower than the mean saliency, to show that basically marmoset monkeys which have a different pattern here, are looking at salient locations. So the high saliency is bigger than low saliency. This is high minus low. Marmoset monkeys are looking at high salient things more when they are further apart. So this is pairwise gaze error, pixels. So this is how far apart. So the videos were only about 600 pixels wide. And so this is actually basically looking at two different parts of the video, right, with high saliency. In contrast, both the macaques red and the humans yellow, we're looking when they're looking at high saliency things, they're basically looking at things, the same thing, right? Things that are within 100 pixels of one another. Whereas when they're looking at things that are lower saliency, they're looking at things that are further apart. And that's the same if you use saliency percentile too, but again, this doesn't correct correctly for the biases of each subject, and so I don't really, I prefer the AUROC. So the interpretation is basically that macaque is very similar to adult, whereas marmoset is similar to what? Something different. I want to remind you that in human infants we saw the low inter-subject correlation, but similar saliency predictivity as with human adults. So to remind you of those plots, here's a plot showing the adult versus infant saliency. And this is the inter-subject correlation, where it's much lower for the young infants than for the adults or the older infants. And so basically we want to argue that this pattern holds up for infants, and it's the same for marmosets, and so we want to maybe for this reason infants like marmosets look to varied but still salient targets in the same way that marmosets do. To summarize, the different studies had many different sort of conclusions, but that may be due to analyses methods that don't really quantify gaze behavior in a great way that's reliable. So we suggest using the model-based sort of AUROC or inter-observer model type things, type analysis. Using our novel analysis, we showed that marmosets look to more varied but still salient targets. And currently we're looking at data from we're collecting data from human infants to sort of verify this hypothesis or show that it's not true. Basically, the idea is the same: head fixed, show Sesame Street very short for a few seconds to draw their attention. They're sitting in an infant seat, a car seat. Then watch our silent videos from the same database with the same rough size as the marmosets, macaques, and humans. And you can see I added this green for infant. They're looking around. This is slowed down 3 times. This is from one infant from one recording session early in the morning after eating. Here's some more examples from the same session. One thing you notice is the infant seems to be making less or fewer eye movements, so sort of sleepily looking at the same place. This might be because, as we've actually found from other sessions, time of day, amount of time since eating, and the mood of the infant have significant effects on things like the intersaccade interval and the irritability of the infant. And so that might be something we need to either average over or correct for it, just choose some state to be in. I know there's previous research showing looking at infant arousal and how that affects eye movements, and this may be related to that. So look forward to that in the near future.